So thanks for inviting me. And it is my honor to share my work with eminent neurosurgeon scientists because, very few neurosurgeons, they are kind of scientists. So, and basically, I'm a translational neuro-oncology researcher working primarily only like primary brain tumor and brain metastasis. And today I'm going to talk about the how to solve neuro-oncology problem in the operating room by intraoperative tissue, my experience in last 4 years. So this work is supported partially by NICO Corporation. So other than that, I don't have any conflict of interest. So in 2015, a USA-based precision medicine initiative was announced through an NICE called All of Us. Its ultimately goal is to develop the precision-driven healthcare for individual and population with greater understanding in the disease biology and pathogenesis. It benefit researcher include to assess 100,000 whole genome sequence, assess 165 genotyping. It also build the collaboration with other researcher. And to develop the precision medicine model, we need very high quality tissue and standard procedure of collecting from the OR and preserving the sample. But we have to know that we are getting the researcher always getting the sample after the maximum portion goes to the pathology. So the timing is important. So neurosurgeons are very important because they can detect the sample and what time it has to go pathology, and they can keep the time, how fast it can go to researcher. So there are numerous challenges in neuro-oncology, up to 20% sample, I think more than that, collected by current methods fail quality control. It is because there are all the neurosurgeon has different way of collecting and sending sample of pathology. So there is not standard protocol they have to follow them. So that is the biggest challenge in neuro-oncology. And without higher quality tissue, extensive research in proteomics and epigenetic is not possible, including the pharmaceutical company is always sometimes. I had a funded grant in Pfizer and Eli Lilly. They want to test the ex vivo slice culture. So, but if the quality is bad, you cannot do the research. And there are several challenges like intertumor heterogeneity, maintenance of tissue architecture, biology, and variability, and multidisciplinary collaboration. So to do a particular research, we need a teamwork. All people should be in the same page. It is not always happening. So you can see in this slide here, glioblastoma, there are 4 subtypes like classical, mesenchymal, proneuronal, neuronal, and each has very different genetic character. Like suppose the surgeon is collecting sample from here, they rarely can get in one group. It may be mixture of 2 or 3 groups. So the target therapy fell here. So this is the biggest challenge because intratumor heterogeneity. And there is a significant variability. Suppose you can see here, I isolate say 5 to 7 colony clone from the one GBM patient. I grow in genome graft. Some grow aggressively, some don't grow at all. And this is the data from the 5 patients of our particular group. We isolated 33 subclone. You can see there are some drugs, the red is they are working very good, but the blue is not working. It's based on Zeta scores. That means we lost the opportunity for drug development because it said the drug response reflect tumor heterogeneity. So that's the problem. And the last one is successful team. In MUC, like no, we already like, I can go back. In MUC already we have formed a group like generally the neurosurgery nurse staff, generally they sign the consent, inform the pathology and lab researcher, the sample is coming, and neurosurgeon are the main person here because they detect, because they will tell the sample which area they're sending, and whether the good sample or bad sample, they have to inform, otherwise, it's difficult. So these 4 people are very important, group are important, like basic researcher, neuro-oncology surgeon, and pathologist. So the next generation sequencing system require exceptionally high quality tissue. You can see the RNA and the protein, they are very delicate. Within 30 minutes, it degrades significantly. if you are giving sample for 2 group and if it is degraded, then the results is drastic, significant variability. So it has to be preserved. So this is very important. So that's why always the neurosurgeon are very important to be involved in this project. So like 3 years back, I started some project with the help of Myriad system just to compare what I'm doing. Like initially, I used to do the standard operating procedure, collect the sample and save it at artificial CSF medium. And this we just compare with APS, the Myriad system, and I save with APS modified medium. The modified medium has all the ingredients which can save the slices up to longer term with ambient CO2. We design the experiment in keeping on my mind. We can send, suppose we are in Charleston, South Carolina, we want to work with somebody in California, that area. We can send the sample and work together. So that's why we designed the media and that one. It is published in World Neurosurgery Journal. So I'm going through that paper data. So you can see here, initially, as I said, that this is a 3-step process. Surgeon gave the tissue, I put in petri dish, slice with mech chopper and uniform size, then transport to the petri dish. And from there, we check the viable or undamaged sample and put it 6-well plate and measure the protein viability assay and for testing the drugs. So pharmaceutical is very interested on doing this one. But now we have a better technique, Myriad, because we directly collect this sample. It's immediately processed. And while doing the experiment and heat trial method, we know if there is a sample while calling, make it sure that there is no blood because it has neutrophil and other stuff, flush it. So that will make this sample more stable. So we have done that. Now we are getting almost 99% of viable sample. So this is a good finding. So from there, we went to the same way. You can see for isolating cells. For cells, you have to take the sample and chop it and put in there. But here directly smaller site, you just want to pet it will grow like that. So for me, it is a very good, like my technique has improved significantly. I am involved in a lot of projects. So you can see the APS from what I was doing before and after having the APS system, we have there are 4 benefits. It drive over efficiency, limit the variability, automatically harvest and preserve, reduce the time and effort, will need less people, right? And second, improve the sample rate and reduce the sampling error. And another thing, one way chopping this one, although it is uniform thickness, but the size sometimes bigger and smaller. But it has the Myriad has a uniform size, uniform area. So drug testing result will be better, right? And we can navigate the collection. So that's why I'm interested. I'm working this one. And last 2 years, this system is with me in animal. We have done a lot of work and we submitted some R01 grant. So let's see what is coming. So this is the result I'm going to talk to you. So this is first. Generally surgeon collect the sample and saline moisture gauge and room temperature and send to the pathology. I'm telling what it is happening in MUSC. So some surgeon does that, and some put some media and saline and do that. And for my lab work 3, I just did collect the sample artificial CSF media. So and the 5, 6, 7, I collect the sample with Myriad and putting in the APS modified media. And this is 3 different temperatures. Suppose 37 degree, why I did? If you are interdepartment collaboration, you can send the sample at 37, and you can ship the sample at room temperature of 4 degrees. Some people like 4 degrees, some people do it room temperature. And that's why we did. And APS has different ingredients. We can preserve the sample for a longer time, and you can close it. So because it has the ambient CO2, right? So that's a good thing. And you can see here, this is the color. You can see 15 minutes, 3 hour, 12 hour, 24 hour, 48, 75. It's degrade this one. The 3 also degrades with ACF medium and degrade. And 4, I just compare collecting the sample from a Myriad system with the ACF media. It is better. You can better, but not better like the modified medium this way. So basically, I'm telling if this result says that the cell can be viable up to 48 hours. And with this medium, and if it's collected by Myriad, and the procedure that we follow in this paper. So you can go next. The same thing, RNA also, it follow the same trend. And if you go next, protein also some, but you can see some degradation in longer time, but it don't take like more than 48 hours is kind of fine within sending the sample with a particular protocol, and we can collaborate with other institute. And live cell, I won't prefer 4-degree because when these stay in the cold, it is very difficult to recover the cells. If it's room temperature around 37, if you send to somebody, it's easy to isolate, but it won't matter because there are few cell lines, it will grow again. But this is the way I work. So basically, other than excellent quality, and my paper work to tell about tissue transfer between the laboratory. So, and it is essential for scientific advancement, and it can send USA within 24 hours from one lab to other laboratory. And for most European researcher, we can try to send 36. It's going within 36 hours is good. And it has maintained, and we have tested, and we are working on a lot of other stuff like metabolic character and other stuff, RNA sequencing. We are doing a long-term study also with more patients, 30 patients. This is the data up to 6 patients. So this is not future. I'm already started. Some of the questions already raised by here, some of the scientists here. So what I'm doing, I have done successfully, collect the sample from OR, put in the, it is immediately collected, right? Within 30 minutes, we can put in humanized mice and it grow. The PDX is grow. So the PDX model, it is better than other cell line model. Why? It has the stroma, this small fragment, it is surrounding by the stroma, right? So it is kind of same environment than what people used to do. So it is more clinical event, but success rate is 20%, although less. But you know what, if one you grow, then that is more clinical relevant the others, right? So, and most of people know that in 2004, FDA introduced a phase 0 clinical trial, a new mechanism to accelerate streamline drug testing for approval process. This is a new approach, unlike conventional phase 1 or 2 or 3 trial. It bridges the gap between the drug testing and define the efficacy study. Its goal is to, like, our team and with Nico combined goal is to quickly identify how the drug works in the patient, whether it should be fast track for further development. So I think this is what we are doing, and we have maybe I'm going to present some work in the SNOW meeting if somebody is coming. So this stuff. And the second, already it was raised by somebody like that we are doing the surgical resection muscle, right? So this is the one instrument you can pass as one to other without sacrificing the mice, right? And when you do that, you can see that if there is some clonal change and thing, you can discard it. But when you take out the sample, the same animal you can use as a surgical resection method, right? This is a good model for study the microenvironment. So this is also going on in my lab. So next. And one of this project I'm working with the neurosurgeon in MUSC and Dr. Frankel and Dr. Vandergrift. So, and we published some paper that a surgeon has the power to take out the tumor whenever they see it, right? So somehow although they take it completely, somehow the tumor arises from subventricle zone, from here to here. So we don't know what is this. So with when they're doing this one surgery, if possible, because they take the different area sample, and we just determine with Myriad system, determine the composition of macrophage and mutants stem cell, non-stem population, subventricle genes, all the population we are measuring this one. And my logic is rational is tumor microenvironment may be responsible for some migration from subventricle zone to this area. So this is the stuff. And a lot of, when I'm telling it is not possible without the Myriad system we have. So my work in last 3 years improved significantly. And the same thing. And the different area, as I said, subventricle 1, 2 zone, I put in mice, right? And if a common, then with a common drug, you can treat. My project is like Avastin, and I'm doing this one and see the clonal chain. If all three area, like if you're taking 3 area and putting in the mice, if drug works, that means this is this best drug for taking to the clinic, right? So, and in this myriad system also, we can check time to time without sacrificing mice, the drug effect. So this is a good stuff. And we made some progress. Hopefully next time I'll have a complete set of data. So finally, I want to tell that always neurosurgeon scientists, that's why I use a neurosurgeon scientist. They're very, very important. And they're basically bridge the gap between discovery science and clinical application. And there are still challenges exist for some dean and hospital CEO, that there are common belief that neurosurgeon to generate to revenue for the institution, emphasizing the role of discovery science for neurosurgery faculty. And always they are busy. There are a lot of clinical workload, and there are declining in reimbursement and as for funding curve. But this is the neurosurgeon are very more important to take. If anything you do, it take to clinic or any type of like now clinical trial is failed now. You can see in glioblastoma there are only 4 drugs, right? Temozolomide radiation and Avastin and TTF field. This how many years? So we need neurosurgeon, more scientists to make it progress. So with that, I'm telling already to build the team. These are the people I work with everybody here, and Dr. Patel, Dr. Patel, and now Dr. Vandergate is working. And we have pediatric neurosurgeon, Ramin Eskandari. We have pathology and Milad radiologist. These are the people who I work together. And I'm grateful for these people, Jomar, Penny, and Heather, and Joe also for bringing together to all the team together. And I always want to collaborate with people, and this is my cell phone number and phone number. You can find here. Always wants to collaborate. Thank you.
Thank you, Dr. Das. A wonderful demonstration of the teamwork and the program you put together. I think your last slide sort of said it all, the issues involved and why we need to do programs like this. One question, do you have any application outside of tumors for your technique and your method?
I think basically primary my focus was the glioblastoma, meningioma, and medulloblastoma, because the company now interested, okay, in my scale, primary brain tumor is not there. So they said that try some brain metastasis model so that we can fund you. So that is I'm talking, but I'm staying in brain tumor, but there are other collab. I don't want to divert what I'm doing agency. I'm to focus, make progress. But there are other groups, they are working on spine, and Dr. Spoeta they are working in ICF and in our department. And I think Nico is already contacted and communicating with them, but that is not my topic to talk, but I'm focused what I'm doing.
Great. Again, congratulations. Questions, Dr. Rutka?
Yeah, thank you for your presentation. I've listened to the last 3 talks with interest, and one of the issues is with the time taken once you do the tissue procurement to go ahead and do some of the studies, like place the tissue into culture. Sometimes it grows, sometimes it doesn't grow. The organoid systems, you have high throughput drug screening on early passage tumor cells. T cells were mentioned as a component of the tissues that you're taking out and so on. So it would be really good for those of you doing this work to do a comparison as to which method of tissue procurement, whether it's the NICO device, the Cavitron, the Sonopet, or the standard procedures that you mentioned, gives you the shortest time to develop, the answers that you need through these various assays. Like how long would it take the NICO versus the Cavitron to get a good organoid model up and running? So, I mean, that's just a challenge, I think, to all of you in this group to look at very carefully and be very important answers for all of us to have.
I think more neurosurgeon scientists. We need more neurosurgeon scientists. So, they will, if they follow the common protocol, it is very rare, but I think this group is helping to bring together. Maybe that is whatever they do, if they follow the one protocol, one way, then it can be reproducible. That's why, glioblastoma, so many years, only 4 drugs, like temozolomide, radiation, Avastin, and now the TTA field. So that's it.